Teclistamab and talquetamab are two bispecific antibodies that have been introduced into the clinic over the past few years. Now, both of them work in a similar way. They both bring the immune cells called T cells close to the myeloma cells so that the immune cells are able to kill the myeloma cells. So in a way, it is dragging those reluctant immune cells closer to the tumor cells. The difference between the two bispecific antibodies, the teclistamab and the talquetamab, is that they target different proteins on the surface of the myeloma cell. Teclistamab targets a protein called BCMA, or a B cell maturation antigen, whereas talquetamab targets another protein called GPRC5D. Given the efficacy of the talquetamab and the teclistamab in patients with relapsed myeloma, a trial was planned to look at the combination of the two. Now, there was an initial phase 1 study that showed really good efficacy in patients with the relapsed myeloma, with efficacy that's almost at par with what we have seen with the CAR T cells. Now, that was the original trial that has already been published, and the combination appears to be quite effective, and it has already started appearing in some guidelines. Now, one of the observations on the phase 1 study was that patients with extramedullary disease, and these are basically myeloma cells that are growing outside the bone marrow, those patients actually responded very well. Now, the challenge with extramedullary disease has been, it has been difficult to treat because the response rates that we get with the current treatments are often lower than what we would see in the setting of myeloma where there is no extramedullary disease. These patients have very aggressive course, and the treatment's effect often don't last, and the myeloma tends to come back faster when patients have extramedullary disease. So it is really an area where much of the progress that has been made in myeloma has not really touched these patients. So given the signal that we saw in the initial study, a separate study was conceived where we only enrolled patients with extramedullary disease. Now, of course, they can also have other disease, but they had to have at least one area of extramedullary disease that is completely separate from the bone disease that we can sometimes can kind of spill over from the bone. And the trial enrolled 90 patients with this pure true extramedullary disease, and treated them with the combination of teclistamab and talquetamab, like what was done in the phase 1 study. Results from the EMD cohort in the RedirecTT study showed that the combination of teclistamab and talquetamab was quite effective. In fact, the response rates were higher than what we saw in the phase 1 study. Now, the unique nature of this trial is that this is the largest trial that has been done looking specifically at the extramedullary disease patients. And we are seeing almost a 80% response rate with the use of teclistamab and talquetamab, the combination given every other week. Now, in addition to the fact that these patients got deep responses, the responses were also durable, with about 60 or so percent of patients still free of disease progression, 9 to 12 months from the start of therapy. So we think the median progression-free survival is in that 12 to 14 month range. And these results are much better than what we have seen with any of the other available agents, including the results from the CAR T. So we think this represents a really good option for patients with extramedullary disease. Yeah, so we did not see any new side effects over and above what we have seen previously with the teclistamab or talquetamab. So we did see hematological toxicity that we see with both teclistamab and talquetamab. There was definitely an increased risk of infections in line with what we saw in the teclistamab treated patient population. And we also saw some of the same dysgeusia, the altered taste, and the skin toxicity that was seen with the talquetamab. So overall, combining the two did not seem to increase the risk of any of these side effects, and we did not see anything new. We still also saw cytokine release syndrome in about 70% of patients, and we also saw the ICANS in about 11-12% of patients as well, like neurological toxicity. So I think the overall toxicity profile does not appear to be much more than what we saw with either of these bispecific antibodies used alone. So I think this is interesting. It's again a single-arm study with 90 patients, but compared to anything that we have seen in the past with patients with extramedullary disease, these results are quite impressive, and the results also seem to be quite durable. So I think this definitely opens up the possibility of using these combinations in patients with EMD. Obviously, it's part of the use of this would hinge on an approval of this combination for these patients.